Good morning, all. This is, I believe, our fifth class. Hopefully you had a good Easter holiday. Today we're going to look at a few things after electrophoresis, which we looked at before. We're going to look at the ideal of southern blotting and hybridization, okay? For southern blotting, the ideal being is now that we have cut up the DNA and got our RFLPs and our PCRs, in the difference between the two, which we should know that RFLPs are very, very long and very, very specific, where PCRs and tandem STRs, they are short tandem repeats, are small, but you can do a lot of them, and therefore you can narrow down the field quite a bit. So between the two of them, that's what they use for southern blotting. To do that, they put that into a process called southern blotting. And for southern blotting, the ideal being is we have to know what we're looking for. So if we have a piece of DNA, we know it belongs to a person. We know that's called a standard reference. We then look at that DNA and say, okay, this has something unique here, 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 and here. Then we pull those things out and we mark those. We mark those with a process called hybridization, which is going to be on slide 24, sorry. And hybridization basically is nothing but attaching a certain type of marker. Usually it's radioactive. It doesn't have to be anymore, but at one time it did, okay? So it usually has to be, something that you can easily pull out and easily make show. They used to use radioactive tags because then all they'd have to do is just use a different color light. Remember our alternate light source, our ALS? They would use an alternate light source to make that glow, and when it would glow, you could automatically see it. You take a picture of it and move on. But you'd have to go through a process of using an X-ray. And by doing the X-ray process, that slows it way down. It's like 2, maybe 3 days to get it all taken care of. So instead of doing a type of thing that uses an X-ray, they started using other types of marker for hybridization, which are not necessarily radioactive. They will react to different types of ALSs. So it just sped up the process. So instead of having a 2, maybe 3 or 4 day process using southern blotting, now you have a process that only takes a few hours, okay? And what happens is the same thing that happens with the radioactive thing that takes 2 days, but it's shorter on the other part. What they do basically is they run it through, they cut it up using the restriction enzymes that match what you're looking for. And then they put it on a sheet, and from that sheet, they shine a light on it. From that light, it glows, and take a picture of it, and they move on, okay? From there, they can put it easily into wherever it is they need to do. A positive RFID test means that you're just looking at and you see a matching band, and the matching band matches up. So you'll see what you need, you'll see what you look at, and they'll match up person to person, place to place, piece to piece, okay? Once you have that, then from there, you can go to the ideal of, well, what if I don't have enough DNA that I can't do RFLP? Well, then you use the PCR testing. Then you do and you just do it faster and faster and more, and you build up until you have an amplified enough quantity, because you can take a little bit of DNA that you find at the crime scene and amplify it and make as much as you want, that you can make copies of it and keep going and going and going until you get how much you need, and then you can start looking for the same thing. Using, instead of the long RFLPs, you're looking now for just a short strand of DNA. Usually that is using the ideals of the STRs, okay? Because everybody has STRs, and those STRs are in different places depending on who you are. So they can basically repeat the process over and over and over again until they get to what they really need, okay? The good thing is that you don't really need that much of a DNA to do it. It's very, very short. Therefore, it makes it very, very quick and very, very fast. And then on top of that, if you only collected just a little bit of DNA, you don't destroy it. You keep having more and more of it. So you're good to go when it comes to having everybody on the prosecution and everybody on the defense being able to do that. The other thing that it does is it doesn't limit what you need. As long as you have enough that you can get some short tandem repeats in a piece of DNA, which is very, very small, okay, it really you know, you don't have to look for a specific gene. You're not looking for anything. You're just looking for certain things that just match up with the other piece of other DNA, which makes the process very quick and very, very individualistic. Hold on. Okay. You can basically see the person no matter what. Now, since everybody has these short tandem repeats, they serve as identification marks. Remember when we did fingerprints? We said that if you had like fingerprints and you were looking for certain markers on your fence or on your fingerprints, you had to have so many of those in order to put it into the system. Well, the same thing happens with STRs, okay? You have to have so many. If you don't have those so many, then it won't work, okay? So the ideal being is that if you have 3 to 7 bases in length and you have those STRs are all 3 to 7 bases in length, and you have enough of those in a certain section of DNA, then you're allowed to put that into CODIS, okay? You must have at least a sample size that will give you a unique combination for that individual. They have determined that for that to happen, you have to have so many STRs that are found in the human genome that are only characteristic to that one individual, okay? And for that to happen, you have to have 13 STRs. If you go to slide 39, you'll see that's what they have to put in there. That's what the government, the law has said. You know, remember when we talked about fingerprints, they said, well, if you had at least 8, that guy is a suspect. If you have at least 12, it's more than likely he did it, okay? If you had 16, it's his fingerprints. Nobody else could match 16. Well, it's the same thing with these STRs. If they have 13, that individual is that individual. There's no way it could be anybody else. And that is what is put into CODIS, is the national database, okay? For all of those people, they really look at the ideal of multiplexing, the ideal of picking up those 13 different areas. And that's why it's called multiplexing, because they're looking for 13 areas or more. If they want to narrow the field down, they can only look for 5 and then take those samples that they get back and then look for more out of that, just like we do with fingerprints. You know, if you run it through the fingerprints profile, they do the same thing. They start off looking for maybe 5 because it narrows down the field fairly quickly. Then from there, they'll go to 10, and then it narrows down the field a little bit more. Then they'll go to 15 and narrow it down, and then they'll do that 16th one, which puts it through, but it's still in the computer, and the computer can do it much faster than a human. Same thing with CODIS. They'll start off with probably somewhere around 7. And then from there, they'll go to, usually they go to 12. When they have 12, then they'll go to the 13th one to make it to the individual. And that is how the computers work fairly quickly, okay? And that gets us to the end of what we're going to talk about for today, okay? But the ideal is that if you use a small amount of DNA, all you need is 125 picograms of DNA for an STR, which is so small, you know, it's so much easier to do because it's 100 times less than what you would need for RFLPs. So, you know, a small amount of DNA is really all you need in order to make this thing work. And it's gotten better and better and better as the years have gone on. So I hope that this gets us going down the right line. Don't forget to watch the video on Southern blotting on Khan Economy, and then answer the questions that I'll put out on Blackboard on the discussion board. Thank you. Bye-bye.